From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 13th, Friday the 13th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 59th episode of Matters Microbial. Thanks so much for listening and or watching and for spreading the good microbial word. It really means a lot to me. This will be a different kind of session, Micronauts. I'm currently visiting Montana State University near a mecca of microbiology, Yellowstone National Park. By the way, here are a few photos of my time in Yellowstone National Park. I had never been here, which is quite shameful for a microbiologist. I hope that I now have my microbiologist card. I'll be chatting with Dr. Brett Payton, Dr. Dana Skorkupa, Dr. Zachary J., and Dr. Anthony Coates, who will all do work with unusual microbes in this unusual and fascinating environment. They all have different roles and perspectives, and I'll be talking with each of them for about 15 to 20 minutes. So let me welcome my guests to the Quality Quorum for today. Hi, Brett.
Hi there.
Would you like to tell everybody what your title is?
Sure. I'm a professor of chemical and biological engineering at Montana State University and also the director of the Thermal Biology Institute here. We focus on Yellowstone and microorganisms that live in the hot springs of Yellowstone.
So I'm staying away from the jokes about some like it hot, but they certainly do.
Yes, they do. We actually have coffee mugs that say that.
I imagine you'd like to start with telling the listeners and viewers a little bit about Yellowstone National Park.
Yeah, I thought I'd do a little bit of a background on Yellowstone and then turn it over to Dana Skorkupa to continue some of that work. So Yellowstone is the result of a huge volcano they call supervolcanoes. It's gone off over millions of years, and approximately the last one was on the order of 630,000 years ago. Yellowstone is the result of a hotspot where magma is coming up to the surface from a very large magma chamber that is underneath Yellowstone National Park and Wyoming, Montana. It's estimated that it's got about the same volume as 11 Grand Canyons full of magma. Yellowstone National Park was established by Congress in 1872. It's up in the top northwest corner of Wyoming, so very remote part of the U.S., and it's the first national park that was ever established in the U.S. and probably the first one throughout the world. It's a very large park. Yellowstone has approximately 3,500 square miles of land area, approximately 800 or almost 9,000 square kilometers. So it's very large, takes a long time to get from one side to the other. The caldera that remains is approximately 30 miles by 40 miles long, and you can see in this map that it's shown and outlined in pink, and it's just a huge caldera. You don't even know you're inside a volcanic caldera when you're there. There's over 10,000 thermal features in Yellowstone, which basically is over half of the world's hot springs, just south of Montana State University. And less than 1% of those hot springs have ever been characterized for their microbial life. So there's 99% of the hot springs out there that nobody's really sampled to even see what organisms are living there.
Brett, may I ask a question?
Sure.
Some of the springs appear to be in the figure acidic, neutral, and alkaline.
Mm-hmm.
Does that have to do with groundwater or geochemistry?
That's basically a result of the groundwater and the geochemistry that the water moves through. A lot of the acidic springs have sulfur, basically sulfur controlling the pH. The alkaline springs typically don't have much sulfur in them.
Thank you.
Sure. I'm representing the Thermal Biology Institute that was established in 1999 to study the microbes in Yellowstone National Park. And so we've been doing this a long time. I'm definitely not the first director of TBI. Over the years, studies of Yellowstone have changed quite a bit. It used to be bears begging for peanut butter and jelly sandwiches at the gates, and now they use scientific studies with satellite tracking for bears. And it's sort of the same thing with the hot springs. For a long time, nobody knew that there was any life in the hot springs, and then people started doing studies and doing enrichments and finding that actually these hot springs are very, very diverse, and that you may have thousands of different organisms living at very high temperatures. And one spring right next to another may be completely different. It might have a completely different microbial community based on the water chemistry, the temperature, pH, things like that.
In earlier episodes of the podcast, we've discussed a lot about how microbes tend to reflect their environment, and I use bad terms a little bit. It's kind of like almost the area in which the organisms live is kind of shaped their genomes. And I would argue, in fact, in some cases, the microbes are altering their environment. That's not germane to this. So it isn't necessarily a surprise to me that springs right next to one another would have very, very different sorts of populations. And it really gives us perspective about the small world around us.
Mm-hmm. We were just sampling last week in the Heart Lake area, and there's a spring that is in about 80 degrees C and pH 8.5, right next to a spring that's around pH 5 and about the same temperature, and just within inches of each other.
I don't know if I've had a chance to tell you this, but for many years where I teach, I used to have students do enrichments from hot water pipes,
mm-hmm.
and we would get thermos.
Mm-hmm.
And then what happened is somebody cleaned the water pipes, and we've had no success since.
Oh, really?
Yeah.
Oh, that's surprising. You'd think they'd recolonize it. Thermos is one of the easier ones to grow when you use typical carbon sources. Thermos comes up pretty regularly. In addition to bacteria, there's all sorts of, there's Archaea, there's each of those organisms has viruses that infect them. And again, that's a very, very understudied area of research in the thermal systems. So thermophiles are typically considered organisms that thrive at extremely high temperatures. And so a moderate thermophile is 50 to 70 degrees C approximately. So if you go turn on your hot water tap, let it get to full heat, that's typically around 60 degrees C. And so that's kind of a cold temperature for most thermophiles. The extreme thermophiles can go up. The highest reported is around 113 degrees C. And so they can grow even above the boiling point of water. Other conditions that these thermophiles can tolerate is toxic metals. They can grow at very low pH, so very acidic conditions, or very high pH, very alkaline conditions. So I want to leave you with why study these thermophilic systems. They're very unique biogeochemical systems where some chemical reactions that wouldn't occur at lower temperatures might be occurring. It also helps us understand the breadth of biodiversity on Earth, can provide clues to conditions on early Earth and evolution. So back when there wasn't much life, a lot of people think that first life was probably a thermophile. Can provide biosignatures for us to look for life on other planets. And then as a bioengineer, the thing that I really think is very cool is only a small percentage of these organisms have ever been grown in a lab. And so there's tremendous biotech potential for thermophiles for society.
I tell my students, in fact, I did so last week, the story of Thermus aquaticus and Tom Brock and Hudson Freeze. And I love that name, Hudson Freeze. That's a supervillain's name,
Yeah,
don't you think?
It is.
And what's so interesting about it is no one had bothered to look. And so this honors undergraduate, Hudson Freeze, was the person who was able to show that what he thought were originally precipitates were in fact thermos growing.
Mm.
And people think, well, okay, that's interesting. And it's more interesting than one might think, because that organism, Thermus aquaticus, all of its enzymes have been adapted to the higher temperatures at which it lives. Mm-hmm. So that means anybody who's done PCR kind of the old school way like me uses an enzyme called Taq polymerase, and the Taq stands for Thermus aquaticus. Mm-hmm. And it completely revolutionized biology. So this idea of bioprospecting, looking for new enzymes or new products that come from these unusual extremophilic environments, is something that is fascinating, is economically important, and as you just pointed out, is a real source of other resources for the future.
And I just want to add, Yellowstone does a very good job of administering research permits and making sure that we collect samples in the most minimal way possible, the most non-intrusive. We're not there collecting large volumes of sample. We take little tiny tubes and then see if we can grow those organisms back in the lab.
And it's important to remember, because there have been incidents where I don't want to name names, individuals might have isolated an interesting organism and really they owe something to the environment or the people in that environment. And that's what the park has done so well, is making sure it's responsible, for lack of a better term, bioprospecting.
Mm-hmm. Yeah. They do a very good job of that.
Yeah. And it's funny because to me, when I try and talk about this issue with others, they'll think that, for example, biofilm is just scum. Mm. Or they'll say, well, those colors that I see, as when I was in New Zealand, the colors that I see in a hot spring, well, that just must be minerals. And sometimes they are.
Sometimes they are. Most of the time, they're microorganisms.
Right. Or the part that got me is there wasn't enough love for microbes in New Zealand when they just said they were hot water algae.
Okay, and I hadn't heard that.
I'm sure there were some there, but right?
In the alkaline systems that we look at a lot, you can actually tell the temperature of the water at certain locations in the spring, because it'll go from nothing really growing to a green photosynthetic organism, and then that transitions to orange and then kind of a brown. And at each of those interfaces, you can tell that the temperature is, from the white to green, it's like 72, 73 degrees C. The other one is around 67 degrees C. So just by looking at the springs in alkaline springs and by knowing the organisms there, you can tell what the temperature is.
I believe the park put out a color wheel.
That was TBI that put out the color wheel, but we donated it to the park for people to do their amateur science.
Oh, I got to get me one of those color wheels, because they're wonderful. The idea is wonderful. And this is something, again, I get very enthusiastic about the microbial world, but I want to remind the listeners and viewers that this is not just about cool microbes, or in this case, hot microbes. This is also about issues that impact biotechnology, that affect engineering, and give us more insights into life on our planet. Brent, could I ask you what the number one thing that sold you on microbiology was?
Oh my gosh. I started on my PhD in the Center for Biofilm Engineering, so studying biofilms, and just not long after I graduated, I had done some projects that didn't have microbes in them, and they were so boring. I swore it off completely, and I've never done another project that didn't have microbes in it. I just love that. I do a lot of bioremediation work.
Yes.
Some looking for degradation of petroleum-based plastics. We're doing biofilms on the International Space Station, trying to control those. It's so exciting to work with microbes.
And so that takes us like from Yellowstone National Park to outer space.
Everything in between.
Thanks so much, Brent.
Thank you.
I'd like to now introduce my next guest, and unfortunately, I mispronounced her name. So this is Dr. Dana Skorupa. How did I do?
Perfect. Nailed it.
So tell everyone what your title is and what you do.
So I'm Dr. Dana Skorupa. I'm research faculty in chemical and biological engineering. And I kind of have a mixed role at our university of doing some course instruction and also hands-on research in Dr. Brent Payton's laboratory group.
So tell some people, like, you have a whole course to talk about.
I do, yeah. I teach a course that's offered through our honors college here at Montana State University, and it's what's called a course-based research experience. Ours is centered around extreme microbiology of Yellowstone, but the course is intended to give students both fundamental knowledge about Yellowstone and its unique microbiology and extreme-loving microorganisms. It's also intended to give students a window into the scientific research process. So students, as part of this course, work on a hands-on research project that's centered around microbes and hot springs in Yellowstone. We travel to Yellowstone on a week-long backcountry sampling trip. Students collect their own samples from hot springs that kind of meet whatever their research objective is. So they're kind of driving their own sampling while we're out in the field. And those are actually the samples that they work with throughout the remainder of the semester. So throughout the 15 weeks, at least in the laboratory course, they're really seeing the scientific process through from start to finish. So from everything from experimental planning, hands-on fieldwork, to gaining technical laboratory skills. Throughout the semester, they, through their experimentation, generate data that they need to analyze, interpret, and figure out what it means and where the path forward kind of is in the research work. And also, we practice a lot of skills related to scientific communication.
You know, a lot of people these days have been doing course-based undergraduate research experiences, and I've done that with students where we look for organisms that make antibiotics from soil or lichen or mosses, and that's fine and that's cool, but we're not on horseback.
So what's being commented on right now is during the remote field sampling component, we actually hire a horsepacking crew to run a drop camp for us. So when we're hiking in into the backcountry, we have to hike 9 miles on that first day. Very nicely, we have mules that are carrying in all of our camping equipment and all of our scientific equipment for us. So the students themselves are not riding on the horses, but all of the heavy gear that they might otherwise have to carry is.
I think it's remarkable. So what kinds of things have the students found that you'd like to talk about?
So a lot of the projects that we've worked on over the first couple iterations of the course have really been focused on research that's ongoing in the Payton Laboratory Group. So it's steeped in actual research projects that we're trying to get grant funding for or kind of put together the final pieces on to solidify a publication. So over the last couple years, it's really focused on trying to find heat-loving microorganisms that can be used in the plastic recycling process. So thinking about finding bio-based ways of recycling plastic waste that we currently don't recycle very much of. So that would be your types 3 through 7 plastic materials. This year, we've kind of shifted and we're trying to find thermophiles that can be used to degrade plant bio-waste. So we accumulate on our planet a lot of byproducts from the agricultural industry, forest industry, food processing, and those are actually really nutrient-rich sources of energy that we could feed to microorganisms and basically convert those plant wastes into a useful product. The current model, the primary target is converting plant biomass waste into fuels like bioethanol.
Oh, it's funny. I do projects with my students where we look at microbes that are attaching themselves to the inside of their reusable water bottles. Mm-hmm. And I've always wondered, am I enriching for things that degrade plastics? Mm. And if I had an SEM, I'm betting that if I were to look at the interior, I would see pits developing with microbes in there, but I haven't done that.
Yes, we've actually, not with the course, but with other research work in the Peyton Laboratory Group, have followed Park Service personnel. So there actually are two National Park Service employees whose sole summer job during the summer is to travel around to different geothermal basins and pick up all of the trash that falls into or off boardwalk into these geothermal areas. And Brent had the great idea that, hey, that's actually a colonizing surface that a microbe that might be able to degrade that product could grow on. So we sampled everything from plastic wrappers to the hats that blow off people's heads when they're walking around the boardwalk area that have plastic in them. We weren't super successful with trying to grow microbes that had kind of initially colonized that surface, but that is something that we have tried.
That's very interesting. And, I just read, and I don't know how overstated this is, but I think it's very important to remember we have to keep these areas as pristine as we can while the public enjoys them. I read, and maybe you have too, this thing at Carlsbad Caverns where someone had left a bag of Cheetos. I'm not being silly or flippant with this. And some of the cave personnel were saying, well, this really alters the environment. And I know that most people looking at that kind of roll their eyes and say, how bad could that be? But each of these environments that you're talking about here in the hot springs, they're unique. And as we heard in the previous interview, some of them can be very close to one another with very different populations. So the impact of humans on these pools are a very big deal. I certainly have heard the scary stories about people being injured in those pools by not following rules. But even without that, the amount of trash, this is really quite interesting.
And I would say too, sometimes I was walking a boardwalk with this summer around Grand Prismatic, and people were drawing in the microbial mats, kind of like you would etch your name into the side of a tree. People were doing that into the microbial mats right off boardwalk. So yes, it can have.
And I have to tell you that more than one person has sent me a photograph of that sign, says do not mark microbial mats, and they make a joke about putting punctuation in because my first name is Mark.
Okay, gotcha.
So no doubt I have to get a picture there.
Yeah.
So tell me a little bit about the outcomes with students.
Yes. So it's really kind of fun. By the end of their 15 weeks, the students again are really kind of driving the focus of their research and seeing this scientific process as I described through from start to finish. And so it's really different from a cookie cutter lab class where every week they're kind of focused on a different topic. Kind of the output from what they did the week before doesn't really connect to the next week's laboratory session. In my course-based research experience, it does. Like we've got to kind of get things right or troubleshoot to be able to generate results that are promising or that we can at least figure out what worked and what didn't. And so typically by the end of the 15-week term, they have some promising enrichments that maybe not everything within their experimental design work, and maybe within each student team, certain things didn't work. But generally, we have some promising cultures that students have been able to cultivate either these plastic degrading microbes that could be degrading plastics, or hopefully this semester, microbes that can grow on a variety of different plant biomass materials. And we continue to work with those in the Peyton lab. So sometimes undergrads in the course kind of transition to working in our lab as undergraduate researchers, and they continue to work on those projects. We've used the data that they've collected as kind of preliminary data for grant proposals to support some of our research objectives and ideas. Yes, we have found successful microbial isolates that can degrade these variety of different material types that we test.
Oh, and they're really, for me as an educator, I don't want to speak for anybody else. I get such a rush. I hate that expression, but it's accurate. When a student who isn't terribly involved, now I don't think this happens necessarily with your cure, but where a student who seems a little bit out of it and they've found an organism from their sample that does something remarkable, and against their will, they become excited by that.
Yeah, there is a sense of discovery by the end of the term of what students have been able to uncover and learn about these systems.
And I would add ownership as well.
Definitely.
Because and I've done it the other way, when you have a series of exercises that you put students through in a class, and sometimes those skills are important. I'm not being all judgy about it. The students come to see them as being hoops to jump through.
Mm-hmm.
And so they don't take it seriously, but they don't know what they're going to find.
Yes. And the thing is, I don't either, right? I kind of open up the semester with that, like neither you nor I know what's going to be successful and where things are going to go. And that's how the actual scientific research process works. Yeah.
So the one further thing I'd ask you to do, if you don't mind, is it's something that you and I know, mm-hmm. which is what is an enrichment culture. But I thought that our listeners and viewers would like to know about that.
Yes. So when we sample these hot springs in the park and bring them back to the lab, it's a microcosm of different bacteria, archaea, potentially eukaryotes, depending on how low the temperature of the spring that they sampled at is. And we're specifically trying to get these thermophiles to grow using a target material as their primary carbon and energy source for growth. Now, there could be a variety of different thermophilic bacteria and archaea that are able to use that material for growth purposes. And so an enrichment is when you're kind of exposing those microbes to just one potential carbon and energy source or source material. And it could be a mixture of different microorganisms that are able to grow in that material. Now, the students do also go downstream and do isolation attempts, mm-hmm. either by trying to isolate on solid media or dilution to extinction. So there's other techniques they put in practice to try and take that mixed community and isolate specific species from that mixture.
It's a critical technique. I do have concerns with enrichment culture because I believe that evolution takes place all the time. Mm-hmm. So you don't necessarily know what you started with. But a good example from my own life is students notice I drink way too much coffee. See what I mean? And what they would do as a joke is, well, it's not a joke. It was an independent project. They would try and isolate caffeine degraders. Uh-huh. And I have a whole story about that I won't take up time on the podcast about. But again, you start with exactly that situation, starting with something that you know as a collection of different things and set up the environmental conditions so that just what you want tends to predominate, which isn't as easy as it sounds because mm-hmm. microbes have their own agenda and it's not ours.
Well, and part of that also is that they live syntrophically. They have to live in environments with other microbial community members. And so when we're trying to isolate individual species of microbes, that's not how they grow in their natural environment. Mm-hmm. And if we're not supplying all of the nutrients, the vitamins, things that they're exchanging with their fellow community members, we're not going to be able to get them into isolation. So sometimes isolation isn't possible with some of our enrichment cultures.
And that's critical. It's a paradigm shift. We're so used to the idea of pure cultures and single colonies, and that's not anything to do with nature.
Mm-hmm. Correct.
This has been, I was going to ask you one more question, I should say.
Okay.
What is the one thing that sold you on microbiology? There must have been one experience.
One thing that really sold me on microbiology is the ability of using microbes to solve different problems or issues, either that are created naturally in our living world or anthropogenically by humans. And so I love the applied aspect of microbiology and using microbes to do good work.
This has been, you can see how fortunate these students are to take a class and with this wonderful microbiologist. So thank you so much for your time.
All right, thank you.
The next guest that I'd like to talk to is Zachary J. Hi, Zach.
How's it going?
Can you tell everybody what your affiliation is?
Sure. I'm a research scientist in the Department of Chemistry and Biochemistry at Montana State University. I'm in a group of Roland Hotzenpischler, who you had on actually a few weeks ago, talking about what his lab does. For me, though, on the other hand, I started working in Yellowstone about 2005, involved with the NSF Research Coordination Network that was started through TBI. And that was a really neat experience because I was able to interact with different researchers, including the park, One of the outcomes of that project was actually making available the thermal inventory online. So a searchable database of all the parameters the park has taken throughout, the measurements that they've taken throughout the Yellowstone geothermal areas. And so with that, we were able to basically coordinate microbiologists, geochemists, geologists, and we held conferences. And one of the projects that came out of that was actually the YMP Metagenome project. And so that was my beginning to looking at or understanding that Yellowstone is a natural laboratory. And as many people have already mentioned, many of the organisms are not cultured. And so therefore, the only way to really study them is to go study them in their natural habitats.
So may I jump in and ask you to define for our listeners and viewers what the term metagenomics means?
Sure. So metagenomics is basically going to a sample, any sample in the environment, extracting all the DNA, sending it off, or doing it in-house, sequencing. Depends on your cost and what what your resources are. Then you get that back, and usually it's all just a bunch of small pieces that you have to assemble together, make longer stretches of DNA called contigs. And then what you can do is use those contigs to identify genes and then infer potential metabolisms of the organisms in there. And so what was really interesting was at the time we did the YMP Metagenome project, there wasn't a lot of microbiology other than, the well-known ones and some isolation studies. And so what we had to decide was, of all these 14,000 features, how are you going to sample individual ones or find the interesting ones? And so what ended up happening was identifying hot springs with more or less unique geochemical sort of conditions. And so as Brent had talked about, right, most of the geochemistries from the temperature comes from the magma underneath. That magma also is releasing a lot of gases, including CO2, sulfide, sometimes hydrogen, sometimes methane. And that then rises to the top and mixes with the surface waters. And then that surface water is then expressed as the hot springs that we see when we go visit. And depending on the type of spring or how the gases interact, you can have what are called vapor-dominated systems. And so those are dominated by CO2 and sulfide gas. And so when the sulfide mixes with the water and then hits the oxygen atmosphere, it becomes oxidized, and you get the formation of low pH systems. And then you also get
it's okay.
No, sorry. You get the low pH systems where you have a lot of sulfide. You can smell sulfide. The other ones are more water dominated, so a lot of the influence is water, and those are called alkaline silicious springs. And those are primarily what you see in the lower Geyser Basin. Old Faithful is an alkaline silicious spring, and those pHs are a little bit higher. And what's neat is even at the surface, you can get separation of springs. So you had mentioned how some springs can be acidic right next to a very basic one, and sometimes those are connected underneath, so that when the water hits near the surface, you get the sulfide and vapors coming off before and leaving behind a more alkaline system. So, for example, Steamboat Geyser and Cistern Spring in North Geyser Basin is a good example of a system that's connected. But one's a higher pH, and the other one's actually lower, and then a lower pH. And you have sulfur, and then, of course, as you can imagine, that overlying geochemistry is basically what defines the predominant microbial community in each spring.
So what's interesting here that I want listeners and viewers to understand is that there are many organisms that simply can't be grown.
So far.
And so there are so many what I call nerd fights going on in science, and this is one of them, right? And it's okay. It's very difficult to cultivate some organisms. How's that?
Yeah.
Sometimes,
absolutely.
by the way, because they need other organisms in concert to grow, which is a very interesting idea and is very commonly seen, but hard to study. What I wanted to remind people of is you mentioned metagenomics, and this enables you to actually reconstruct the genomes of organisms that you cannot cultivate. And again, this requires not simply the idea of DNA sequencings, but computers to create the overlaps to create this. That's too many creates in a sentence. But the point is you're able to regenerate that whole genome, and it's revolutionized the way that we look at things. And it becomes cheaper and cheaper every year. Why this is relevant is when you have these interconnected environments, You can have a situation where you ask, why is area A so different than area B? And if you're just looking at a microscope, that won't tell you very much. If you're streaking on a plate, I know you're not, but that won't tell you very much. But the metagenomic approach can, and that can show you how the communities change. And that's what I think is so powerful and interesting about what you're doing.
It's very powerful. It should be understood that we don't recover usually whole genomes. So metagenomes are very fragmented, and it does take a good amount of effort and analysis to kind of tease apart, the potential. And then, that potential, then you can develop hypotheses to test for those springs. And so one of the things so I mentioned the vapor-dominated and water-dominated systems is at the surface you get a lot of springs that are lower pH and a lot that are higher pH. And if you go to these different systems and sample for the DNA and sequence, you get very different communities. And what we find is pH is one of the main controllers, of course, in addition to temperature, that controls the community members in those hot springs. Another important parameter is oxygen, that you can imagine. As well as sulfide. So usually sulfide and oxygen are not usually found together in large amounts. So it's either sulfide or oxygen. And you can imagine those are highly reactive, and organisms in those springs, especially the more abundant ones or the ones that we find more active, can interact with those and actually use them for energy. If you add flow to a hot spring, that changes the community. In fact, there's actually amazing phenotype. We call them these streamer communities or filamentous biofilms. And you can see these with your eye in the springs and the outflow channels because one end's usually attached to like a solid substrate. And the rest of it is just this slimy goo that strings off of through the flow. And this is a microbial community. And what we find is it's actually quite simple in the sense that there's probably 10 organisms in there.
Interesting.
Yes. And so, you have a bacterium, Thermocrinus. And actually what we found in metagenomics is that's a group called the Aquifocales. And the Aquifocales separate by class name based on basically the pH of the system they're found.
Oh.
So, for example, in the more acidic streamers of Norris, you find what are called hydrogen baculum. And then the higher pH, you get Thermocrinus, and kind of in between is sulfury hydrogenemia. You get some cool names that are associated with that.
I've always felt that. And one of the things that I want to emphasize when we're talking to folks about this is that folks who are a little bit experienced with microbiology, you call them intermediate micronauts for lack of a better term, they're so used to the idea of bacteria just having three shapes. First off, that's not true, but we're used to the idea of spheres, rods, and spirals, yes. And there are variations among the bacteria, but when we get into the Archaea, especially Archaea in unusual environments, you get some funky new shapes.
And you see those when you make those geochemical conditions more extreme. Like raise the temperature, you're going to get more Archaea, and then those Archaea express some very different sort of characters. And if you lower the temperature, where now you might say below 73, where you have photosynthesis, you now have a more diverse community who might actually provide more carbon. And so then the Archaea are different in that system as well, even if they're not more predominant. And not surprisingly, in the more extreme ones, we get what are called chemotrophic habitats because there's no oxygen. And most of those are archaeal. And the Archaea are actually utilizing the spring's predominant geochemical parameters for energy and as well as for biomass growth.
And their input carbon.
Yes, right. And we find actually as you increase carbon, the dissolved carbon in the hot springs, you increase the diversity of organisms in there.
Yeah.
So you do almost have like a hierarchical sort of parameters you can choose, but then once you lower temperature, it gets kind of overwhelming sometimes.
I'm not going to put you on the spot, but I guess I am. So the less complicated the assembly, the more extreme the environment?
So, yeah, well, actually kind of the reverse. So
okay.
If you go to a more extreme environment, really high temperature, really low or high pH, the community is constrained. There's not very many organisms that can live in really acidic conditions.
Sure.
So that constrains the community composition and becomes less complex than if you had like a lower temperature.
Okay.
And what's nice about that, especially at the, when metagenomics were taking off in Yellowstone, is that you recover more sequence of the dominant organisms. And so you have a better picture as well as maybe a more complete genome representation of the community members. Compare that to a soil sample, and it's impossible to pull out and make sense of it because the diversity is just so incredible compared to an extreme environment.
And thinking about the human gut, exactly the same idea. It's like the difference between, and I hate how this sounds, the Sonoran Desert and the Amazon rainforest.
Yeah, totally
right?
different. Exactly.
Yeah,
right. Yep.
Very different.
Yep. And then when you go down there, you measure the geochemistry because you want to understand the microbes with the geochemistry there and how it might be different. What makes particular soils different.
So I'm wondering if you wanted to give some examples of some of the organisms that you found, because they have some interesting shapes.
Sure. So some shapes, we focus mostly on Archaea, and Archaea can be anywhere from long filamentous cells to round cocci to various forms of rods. And what we find is that certain Archaea, they might have crazy thick cell walls. It's almost like every archaeum has some unique attribute that we've never seen before almost. I think Anthony will talk about some of that too that we found more recently. But the ones we find are mostly, we were focused on the archaeal system. So we find organisms that are in the thermoproteales, although the name's now changed, because a lot of these organisms originally were the ones that were found that define the archaeal space and in the tree. Since then, that tree's expanded and we find them everywhere. But at the time, it was mostly the thermophilic or the pH extreme Archaea. And so we find, for example, in the streamer systems, there's a pyrobaculum group. And we find this pyrobaculum not just in streamers, but we find them all over the place at circumneutral to basic pH systems that are above 65 degrees C. So we see those, and what's neat about those is when we look at the genomes, we can also bring them into culture, but when we look at the metagenomes, we find that in one system they may have oxygen respiring capabilities.
Right.
While in a more sulfidic system where there's no oxygen, they might be interacting with the sulfur. So their metabolisms change based on the predominant system, and that's probably what makes them so cosmopolitan.
Yeah. So what does pyrobaculum look like?
It's a rod. It can also have different shapes depending on the species too. Some can have like a club shape, like an end that looks like a golf club. They can be branched, but most of them are rods or filaments in shape.
It's not relevant to you right now, but I believe it's pyridictium that makes like a network.
Yeah, so that's I think a desulfurocalcalis,
right,
whips, which we do find also. Yes.
So in other words,
those are crazy.
Right.
Also, they have some weird cell shapes.
Right. And I want to emphasize for listeners and viewers, it is not always simple to grow these organisms if they can be grown in culture. And this is kind of different than with a lot of bacteria, and you start to get this into your head that bacteria are easy to grow. Therefore, Archaea, which are also tiny cells, they're easy to grow as well. And it's just not true. Now, there are some that are easier than others, and I really support that wish. What we need is the E. coli of the Archaea.
Many grad students agree with you completely. When it takes 80 days to grow some of these, it's a long time.
So it's very gratifying when you get the results that you do.
Absolutely. Yes, definitely.
So this has been quite an interesting bit of research. Do you have some take-home lessons for the listeners and viewers?
Sure. So we basically have defined more or less 3 different environments. Granted, every single spring is different, and we haven't sampled all of them, but you basically have 3 different types, the streamer types that I talked about, the chemotrophic sites, and those seem to be more involved with geochemistry. For example, in Norris, the pH is so low, the iron doesn't usually rust. It doesn't oxidize and fall out of solution. So in those systems, we find that the microbes are entirely responsible for iron deposition. So if you go to Norris and you see iron mats, which might also look like phototrophic mats, but the iron mats, that's all microbial action and metabolism. Wow. Right. And Archaea specifically, some bacteria.
So you have an image here of chemotrophic communities. And what I find so interesting here is you're talking about the temperature and the pH and the name of the spring. This emphasizes what we've heard several times today, that you can have very different communities in different springs.
Absolutely. And some of those, like Joseph Cote, has a really high amount of arsenic. We find that organisms living in there can utilize that arsenic for energy, and then also creating sort of a cycle in that hot spring, which then complicates in the geochemistry of the upwelling water from deeper down.
And what sounds great to me is I don't see this research ending.
Oh, no way. No, it's continued. it keeps getting more and more exciting. Also,
Right.
the more we learn, more people are doing it. We're starting to learn from the metagenomes, bring samples back, and then target culture them. And that's one of the things we've been able to do more recently is bring uncultivated organisms into culture. And then finally, we were able to in the phototrophic mat. So these will be a little bit lower temperature than the chemotrophic systems. Is that? And these are more complex and involve more bacteria and that utilize sunlight for energy. But not all of them produce oxygen. Some do, which can complicate things in a spring if oxygen is diffusing in. But also like mushroom spring, that's actually where Thermus aquaticus was originally isolated.
Right.
There. So long-term studies have been associated with the springs we've been studying.
This is so fun for me because though I've never been to Yellowstone, at the time I'm recording this, tomorrow I will be.
Yes.
I've heard about these springs, and that's what's so exciting. So you see names like Fairy Geyser and Chocolate Pots,
right,
and Mushroom Springs. And I've read about these from for many years, and so it's a really exciting time.
It's great to see those. Yeah, and they have been for a long time. In fact, a lot of the people who were involved in the RCN, Dave Ward, Don Bryant, Chris Klatt, they've been studying these longer than the archaeal system.
Yeah, how about that?
It's been very neat. And then I guess I could finish. You asked a little bit about metagenomics, right? And so it's like, okay, well, we assemble all these sequences into longer sequences that are still fragmented. How can we tell what they are? And what we can do is separate sequence into a three-dimensional space. And so sequence, in this, for example, in this plot, each dot represents a contiguous sequence. And the character of that sequence, say the sequence of ATCGs, how the bases are, you can simplify that into numbers and then project them in the 3D space. And so sequences that are similar or different will group together. And so sequences that group together, then you could go in and identify them, and usually that corresponds with a certain taxonomic level, depending on the site. And then, for example, this one shows conch and octopus. So if you have two different metagenomes that are assembled and analyze it this way, you can then compare, okay, which ones are there in both systems, like Thermocrinus, they're the predominant ones, and then which ones are different, right? And a lot of, in this case, the ones that are found in octopus and not in conch are more aerobic. They have genes encoded that allow them to respire oxygen.
So they're kind of diversifying based on the environmental conditions,
Absolutely,
which is
yes.
really interesting.
Yes, yeah. So that's how we're able to study these without bringing them into culture, even though, the impetus is to see what we can bring into the lab.
So Zach, on some recent episodes of Matters Microbial, we've discussed how sometimes we can see evidence of early parts of Earth's history by looking at the metabolites that bacteria have left behind. When I look at some of these hot spring communities, I wonder, is there any intersection with that?
Sure, yeah. So presumably a lot of the hot springs in Yellowstone might reflect conditions of early Earth. And so those conditions would then promote the growth of similar organisms. So the extant organisms we study today might reflect potential metabolisms that were occurring at early Earth conditions where it may have been more volcanic and there was more geothermal input.
This has been absolutely fabulous. I've learned a lot. I'm looking at these wonderful images, and I hate, you've heard me do this with other speakers. Is there one particular observation that hooked you?
Oh man, well, if you say in general, I went into microbiology immediately in undergrad. So in high school, I was actually working in the hospital. So I've been doing micro a long time. But the environment, the ability to go sample and go into the field, and take all sorts of measurements, that was kind of a big hook for me. And
I would argue, and I hate how this sounds, I don't call myself old, I call myself entropically challenged. And what I would say is that even I have gotten to see how the technologies have improved, enabling us to ask and answer questions that we never could have thought of before. And it seems to me like the pace is only growing faster.
It is. It's going so fast. As you've seen, just discovery of Archaea is just kind of going crazy. It's fascinating to watch, and be interested to see where it goes.
And I tell my students that this is why, to some degree, having some abilities in the area of bioinformatics is really becoming absolutely necessary.
It really is. If you're studying actually any system,
Mm-hmm.
understanding how to computationally analyze sequence, metabolites, expression data, you have to know how to use computers to analyze the results.
So my final comment to you is that, are you aware of the essentially pH 0 biofilms near Berkeley?
Yeah, the Iron Mountain ones?
Uh-huh.
Yeah, oh yeah, that's one of the original metagenomics sites, that in the Sargasso Sea.
So they hang from the ceiling in biofilms, and they're called, and I apologize in advance to listeners and viewers, they're called snotites.
Just snotites, yes.
Because they hang from the ceiling, I guess. And apparently they have such low pH to them, you have to wear special equipment when you go in there.
Yeah.
It's awesome.
It is awesome.
And so people think of microbiology, oh, I'm working in a hospital, oh, I'm looking at Petri plates. No, sometimes you're kind of a microbial Indiana Jones.
That's a good way of looking at it. I need the hat then. See, I need the hat.
You need the hat for that.
Yes, right.
Thank you so much for your time. Last but not least is going to be my guest here today, Anthony Coates. And what I'd like to do, Anthony, is please tell everyone what your affiliation and experience is.
All right. Yes. Hello. I'm Anthony Coates. I'm a recent graduate from Montana State University, and I'm now a postdoctoral scholar in the Hudson-Fischler Lab, where I've been working on. So we've been talking a lot about these uncultivated microbes, and my work has been to try to bring some of those into the lab and cultivate them and study them in more detail. And so I've been fortunate in the Hudson-Fischler Lab to have access to sample sites in Yellowstone. As we've heard, there's an incredible amount of features in Yellowstone in such a small area. And in terms of, so I'm focused on Archaea. Yellowstone also has like a rich history of studying Archaea and just finding new and interesting lineages there.
I noticed that you said something in this slide that I'm looking at that I'm fascinated by. Many archaeal lineages were first described in Yellowstone National Park, and my eye goes instantly to the Odin Archaeota and the core Archaeota, by the way.
Yes, yeah. So the core Archaeota are actually one that we've been able to culture, and I'll talk about a little bit later. And then the Odin Archaeota are part of this larger group called the Asgard Archaeota, which people suggest they are distant relatives.
Yes.
And so, yeah, I think that's one exciting thing about hot springs is that you can find these really unique groups, and maybe because they're so undersampled, this is where you first find them. But when you look into other samples, you can see that they're more widely distributed because hot springs are a more simplified community. They can be easier to study.
It's wonderful.
In these areas to start with.
Yeah, and I like how you're balancing culture independent and culture dependent approaches.
Yes. So if I can, I have a story of 3 lineages of methylotrophic Archaea. And so, why do we care about methylotrophic Archaea, and what does that really entail? And so, methyl compounds are ubiquitous. So as you can see on this slide, I have just a few small samples of different chemical compounds that contain methyl groups. And so it's a very simple chemical group. You find it on so many different things, and so you can allow a lot of different, a lot of diversity of microbes can access these methyl groups for carbon and energy. And we can see that they're present in both human-engineered environments like anaerobic digesters, wastewater treatment, oil fields, but also in just really broad environmental samples such as soils, hot springs, and lakes. And so methylotrophy kind of fits into this much larger picture of the global carbon cycle. And so you can have degradation of more complex polymers into simpler amino acid, fatty acids, and sugars that are then degraded into organic acids, methylated compounds, and alcohols, among others. And so these methylotropes, they're able to use sources of energy such as methanol or methylamines and you can have methanogens that can convert this to methane. And you can also have non-methanogenic methylotrophy where you're converting this to CO2. And so methane and CO2 obviously are very important greenhouse gases.
Yes.
And very important for the global carbon cycle. And so they're kind of involved in this final step of organic matter degradation. And then additionally, other microbes can feed on these gases, these methane and CO2 that's produced. And so they kind of intersect as well with other nitrogen, sulfur, and other metal cycles.
You know, there's an old poem, I think it's John Donne, who said, "No man is an island." And I would argue that very few microbes are in and of themselves enough. They so many times work with other microbes. And that's what you're showing right here, and I really appreciate that.
Yes. Yeah. So, I work with enrichment cultures, which we mentioned earlier. And so I haven't been able to get an isolate yet, but it's interesting with these enrichment cultures, you can kind of just see maybe not exactly what's happening in the environment, but you can see some of the like basic interactions.
Sure.
So you can see exchanges of hydrogen between different microbes in your enrichment culture. And then later try to go back into the environment and kind of test some hypotheses of who's working together.
Yeah, and
who's friends.
Yes, who's friends. That's good because they're reducing equivalents back and forth. And it's important to remember sometimes people think that when you're doing this kind of microbiology, you're just observational, even if you're just looking at A, T's, G's, and C's. But in fact, you're exploring hypotheses.
Yes.
And it's important to remember that.
Mm-hmm. Yeah. And so it just talked about metagenomics. And so a lot of the times this, it allows you to get a window into the potential of these uncultivated microbes. So the first methylotrophic microbe I'd like to talk about are called these Culex Archaea. And so you can see on these slides, they are globally distributed in geothermal environments. So not only Yellowstone, which you can see on this map, and these pictures of hot springs, but also other hot springs across the United States, China, and New Zealand. So we were able to collect 10 metagenome assembled genomes from 6 different samples. But we also see through gene surveys that they're widespread in other environments. And so they're only detected in high temperature environments above 50 degrees. And they're often low abundance populations. And this is we assume this is why maybe they were overlooked beforehand, but that doesn't mean that they're not important in carbon cycling in these systems.
No, and this is something that's really important. We tend to look for the low-hanging fruit, for lack of a better term, the easy to isolate, the most numerous, and those low abundance populations can, as you say, be very important.
Mm-hmm. Yeah. And so if you're in a lab and you find a new genome, and it's not really related to anything else that you've seen before, where do you start? And unfortunately for this microbe, it was found to encode a very diverse set of potential metabolisms. And so it has hydrogenases to interconvert protons and molecular hydrogen. It has methyltransferases to enable a potentially methylotrophic lifestyle. It has lots of potentially electron bifurcating complexes, so there's some very strange bioenergetics going on in this organism. And then also just the ability for degrading carbon monoxide, fatty acids, peptides, and sugars. So we're thinking that this is a very generalist microorganism.
It's not fussy.
Yeah. And so because this was a metagenomic-driven study, these are now hypotheses that we can test. So we can take samples where we find this microbe, incubate it with the substrates that we predict that it can use, and see if we could enrich it in the future.
Well, right. So, for example, if you see that they appear to have genes, as in that slide for fructose and galactose utilization, if you spike your enrichment cultures with those particular monosaccharides, you could potentially increase the percentage that would have this.
Exactly.
Yeah.
Metagenomics is often where we start in designing new cultivation campaigns. And so the last two groups of microbes I'd like to talk about involves methane. So methanogens are anaerobic archaea that are able to produce methane and couple this to energy conservation. And so far, we don't find this in bacteria, so it's a very archaeal-specific metabolism.
I like how you said so far.
So far, yes.
Because we all have to say so far
yes.
all the time in microbiology, don't we?
Yes, you have to hedge your bets pretty hard.
Every time. And I love how you're talking about the effect of methane as a greenhouse gas. I've taught many times to students it's more potent than CO2. The number you have here is 28 times, more than 28 times. I used to say 50, and I might have been exaggerating.
Yeah, I think it depends on the model you use. But yeah, we're definitely seeing that atmospheric methane is rising. And so it's estimated that global methane emissions are on the order of 500 to 600 teragrams per year, that's a lot of zeros. And so methanogens are really important in this process because they contribute about 70% of that methane per year. And when you look at the contributions of methane to warming, it's estimated that methane has produced or contributed up to about 30% of the contributions to warming.
That's amazing. And methanogens appear to be making most of the methane being emitted at this time.
Yes. Yeah. So we really want to understand the biochemistry of this process, but also the diversity of this process. And so when we look at the tree of Archaea, we see that the methanogen diversity is being rapidly updated. And so basically, for the last almost a century, it was thought that all methanogens belong to a single phylum called the Archaea. But then when people started sequencing more and more samples in the last decade, it's been found that the key enzymes for methanogenesis are found in other phyla. But the problem is all of those were uncultivated.
Right.
And so we didn't really know what they were doing, and there was no experimental evidence of methanogenesis by these non-euryarchaeal organisms. And many of them were thought to be methylotrophic. And so basically, in my PhD work, I was able to obtain two methanogens outside of these traditional Euryarchaeota, and so bringing in two new classes of previously uncultured Archaea.
Speaking of hedging your bets, right? You wouldn't just want to look at one just in case, right?
Yeah, let's get a duplicate here. And so you can see these are both in the phylum Thermoproteota. So we have the class Archaeota, which we mentioned earlier, were first kind of found in the 1990s. And that is Methanodisulfocora washburnensis. So
that's a mouthful.
Yes, yeah. So it has genes for methane and sulfur metabolism. And washburnensis, it was first found in Washburn hot springs in Yellowstone National Park. And so you can see the hot springs here, and we see that the Archaea are these long filamentous organisms in red. And so we were able to cultivate these at 77 degrees Celsius, pH 6.5, in the lab stably. And so in the bottom left, we see this metabolic model. And on the bottom right, we're able to see what genes this microbe is expressing during methanogenesis. And so we can understand more about it when we study it in the lab. And then on the right, we have this other class called Methanomethylacea. If you're involved in the space, you may also know them as Virology Archaeota. Archaeal taxonomy is a very rapidly changing space. But this species is Methanosphericola virstradii, and we see there are these small little spheres in yellow. And we're able to take a similar approach where we can reconstruct their metabolic model and also see the expression of those genes in culture and verify that these two organisms are in fact methanogens. And were previously uncultivated.
You did this.
Yes.
Don't you feel good about that?
It's nice. I wish they grew a little faster, but it's all right. I'll give them props for being unique enough, interesting enough.
So it's something I think that we talked about before is this idea of having ownership of what you've done. So what's that feel like to you when you've been part of being able to isolate and grow an organism that hasn't been grown before? I mean, are you just like, oh, I finally did it, or are you excited?
Yeah, I mean, I feel like at the end it's maybe Frodo at the end of his journey, but I think
That was nice.
Yeah, the very beginning. So I think the moment that's most memorable to me is so what we're seeing in these images is called fluorescence in situ hybridization. We're able to introduce a specific probe with a fluorescent label,
right,
to specifically target this organism. And so this can tell you whether or not in your enrichment culture these organisms that no one has ever seen before under a microscope, are they present or not? And so the first time I did fish and saw these spheres, I was, yes, I finally have them.
And see, this is what's so important here is that when you look at things in pure culture, you know what you should get anyway. But when you're looking at an environmental sample, there's so much going on, and using fluorescence in situ hybridization allows you, without growing the organism, to show that it's there and sometimes even get some degree of enumeration, which is really fabulous to think about. I think you've just completed your PhD and you're doing a short postdoctoral stint now. Do you know where you're going next?
Yes, so I'll continue to be studying on these methanogenic organisms, but trying to look more closely at their enzymes from a biochemical or structural perspective.
And again, for people who think that this is okay, I like pure research myself, but I find that many people want to know about things that are of use to the everyday world. And I would argue that your work is very important because of the issue of methanogenesis.
Yes. Yeah. So because we only knew that, or previously thought that methanogens were only in these traditional Archaea, we don't necessarily have to revise like the models of how much methane is being produced, but we have to revise our models of who is producing the methane, and to what extent these thermoproteota methanogens are involved in that process.
You know, one of the challenges I think in taxonomy, when you look at the microbial world, is the sheer level of horizontal gene transfer that takes place. When I say this joke in class, I'm only partly being flippant. It's like you having offspring with your Caesar salad. That and again that might take away from the seriousness of it, but it's important to remember that the ideas that we have of isolation between genus and species and animals and plants, it's completely blurred in the microbial world. And that's what you're showing here. It's easy to say you find these characteristics only in this one taxon, but you have evidence that's not the case, which is wonderful.
Yes, yeah, and so I think going forward, since we kind of had this lens, Archaea methanogens have been cultured for decades now, and there's amazing studies of the biochemistry, the structure, the physiology, and genetics of these systems. So we kind of view methanogenesis through this lens, and now with these very divergent cultures, we can kind of look and see where does the biochemistry for methanogenesis start to differ between these groups. And so it opens up kind of fundamental research on methanogenesis and it's like the diversity of enzymes that enable that process.
Well, it sounds like a wonderful thing. Congratulations and best of luck to you.
Thank you.
So I'm really grateful to all of my guests for today. And what I wanted to say here is that it's been a real pleasure to be here, and I'm grateful to all of them. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, comments, or suggestions to me at mattersmicrobial@gmail.com. Show notes from today's episode with epic levels of links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. I want to again thank all four of my guests who are associated with Montana State University and the Thermal Biology Institute, which is an epic name, here in Bozeman, Montana. Many thanks to the ever-patient David Renata for superb editing under challenge, and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.